The theme over here is found in translation around innovations in global health didn't come naturally, so I'll have to explain that. One of the things I'd say in terms of the work we've done with Scripps and Caliber, and I could repeat that many times, is that beyond the excellent science and obvious excellence in science, it has been the commitment to public and global health and to patients, which has tremendously impressed me. And your joint efforts in that regard have been tremendously appreciated. So when we think about this demography being destiny, it brings us to a framing of the global health situation as I wanted to put it to you today. You can think of two major waves of progress in the last 50 to 60 years, with a very large number of people coming out of poverty and this fantastic halving of child mortality. And that began in South America, and it overlapped with the same trends in Asia. What remains? Well, I would love to see the third wave of progress, and I think that that is going to be focused on sub-Saharan Africa for a couple of reasons. If you look at the projections of poverty, which by 2050 we can expect the majority of the world's extremely poor people to be living in sub-Saharan Africa. And not only that, with the relatively intractable total fertility rate, we can expect almost 1 billion young people in that same region. Now, normally you would say that having a large, young, vibrant population would be a tremendous factor in economic success and in improvement in the continent. But I think unless we actually are able to make some of these technologies, that may not be entirely true. And that's what I wanted to discuss today. I've listed here some of the key areas that we work in, but behind this, there are obviously many other factors, whether it be nutritional, whether it be sanitation, whether it be agricultural development, which hide behind these. Today, let's focus on infectious disease as the principal topic. So the one thing I have to confess is when I started at Gates coming out of Novartis, I had a very naive view, as it turns out in retrospect, of what we should be doing. You really need to have simple solutions. So we would work on things like better vaccination cards that kids could keep and they would get vaccinated. And we'd have simple lateral flow tests that you could do in a L0 that's right out in the community kind of clinic. And then we would do, screening of antenatal clinics, simple weighing of babies. So I had this notion that really you're going to take these simple kinds of interventions that we know have been so impactful everywhere else, and you would just transplant them and improve them in some way. Well, the problem was that I underestimated the tremendous lack of infrastructure in many of the regions, whether you're talking about northern Nigeria or you're talking about parts of conflict zones in Democratic Republic of Congo. The lack of healthcare technology is truly amazing. You talk about a physician. Many people will never see a nurse or even a semi-qualified healthcare worker in their entire lives. So when you're talking about vaccination cards, even in regions where getting to the third DTP vaccination is at 5%, then you have to rethink the simple approach is always going to work. And I, over the couple of years working at Gates, have come to feel that in fact, transcending this infrastructure gap requires not just simple solutions, but requires the highest level of technology and innovation, both technology, financial, and social innovation that we can bring to the problem. So I wanted to walk through a couple of examples to demonstrate what I mean by that in some of these areas. Let's take vaccines. The problem we have, this is now the leading cause, if we take out the fact that the pneumococcal vaccine is being relatively widely distributed, and we have some successful pneumococcal vaccines. Respiratory syncytial virus is one of the leading causes of pulmonary problems, death in kids, particularly young kids. So you can say, well, let's develop a vaccine. It's been very difficult. As you know, there have been many efforts to develop an RSV vaccine, and there have been some singular failures. And we're now in the next hopeful phase and waiting. In a fairly short period of time, we'll see some definitive data on the RSV vaccine. But the problem is, how do we capture the kids in their first month of life when birth is often not in a clinic and birth is often at home? So the vaccine that we need is a vaccine that can be delivered to the mother in the third trimester, and we rely then on antibody transfer across the placenta in order to protect the child in those early days, even if the mother never presents to a birth clinic. So now you think about that in terms of the safety burden that you've raised now in terms of the vaccination of pregnant women, the need to have the antibodies be sufficiently potent that they can span many months and cross the placenta effectively. You raise that technology barrier very substantially. What about diagnostics? So there's been a huge investment in diagnostics, and I think we are really on the cusp of having some transformative new solutions in this space. And as much as at Gates we've invested heavily in this area, we have not committed to a platform. At one time, Bill was a fan of what he called the box. So he wanted a box that would do all format of diagnostics, molecular, ELISA tests, all in one box, and you could just plug somebody into that box and it would give you the result. And I think we invested a lot of money in that enterprise, and I would say even as of 2 or 3 years ago, I was pretty dismal about the prospects for having the box. Things have changed, and what is the goal with these diagnostics? What is that barrier that we need to transcend? And I've just put it down as 10-10-10 because in global health people like to come up with these numbers. 90-90-90 is a number formula which is common in the HIV area, and then was 0 by 2015. So they like these things. What is 10-10-10? Well, 10-10-10 is the test has to be available in 10 minutes with 10 microliters of blood. That's a finger stick, not a draw. And it needs to cost less than 10 cents. And as I say, up until a couple of years ago, I would have been fairly pessimistic that a box that could do this would be available. But in the last year even, I think there have been some real advances. One of the ones that I'm very excited about has got something which is getting very close to that barrier. They've launched in Europe and deployment in Africa, and they will be launching at some point in the US as well. So I think that in this space, we see some line of sight to the 10-10-10, which is very exciting. Nothing could be more important for us than surveillance. We've faced a fairly dire situation with malaria in the last couple of years, which is that the tremendous decrease we saw in malaria deaths and cases looks like it's plateaued. And once again, the reason is because of the situation in sub-Saharan Africa. Two countries, Nigeria, particularly in the north, and DRC, are the main contributors to this plateauing of malaria deaths at about 500,000 a year, majority of them in that region. Well, we have the Global Fund, which has been a very important multilateral mechanism for funding the basic commodities to tackle malaria, whether it be indoor residual spraying or it be bed nets. And the bed net problem we have, this is part of the reason for the plateauing, is that pyrethroid resistance to the pyrethroid nets has spread widely in Africa. We don't have a good idea of the exact geography, but there is widespread pyrethroid resistance. We have some new nets which are coming in, which have got dual insecticides, which are able to overcome the pyrethroid resistance. At least one of the components is a non-pyrethroid. we have about 10 million of them, for instance, for this year, whereas the total bed net distribution is almost 100 million. So we've got very few of these pyrethroid-resistant nets that transcend the pyrethroid resistance problem. So what are we going to do? Well, Peter Sands has told us, if you can tell me where these hotspots of pyrethroid resistance are so that I can target those 10 million very special expensive nets, I can double our impact of the Global Fund in terms of reducing malaria deaths. So what we need is surveillance. This is understanding whether we have pyrethroid resistance of mosquitoes within a particular geography. So we need a molecular surveillance. And what form does that take? So here is a proposal. Over here, we have a form of molecular surveillance. This is actually a picture from the polio teams, the WHO and the UNICEF polio teams in Afghanistan. And you see that the two polio workers over there are actually sampling from from a sewer, from an open sewer. This is environmental sampling, which has been absolutely critical to understand the distribution of polio cases. Many people believe that polio would never have been eliminated in India, for instance, without this kind of intensive environmental sampling. And we hope that that's going to have the same effect now in the reservoirs of polio, which are in Pakistan and in Afghanistan. They would then look at, with a PCR method, for the polio viruses that they find in the effluent, and they would be able to understand the sequences to the extent that they would understand where these have been moving in the migration of people, which is so important in all of these situations. And then you have this, which actually is a photo from one of the latest and greatest fads. It's not a fad, I think it's a tremendous technology, which is the liquid biopsy that people are proposing as a way of early diagnosis of cancer, for instance, and potentially other diseases, but cancer has been leading the way in terms of a small volume, relatively small volume of blood, where you can sequence the host factors and you can understand whether there are cancer-related mutations in the sample. So what has that got to do with this? Well, I would suggest that we can think of environmental sampling as the liquid biopsy of public health. The problem with the molecular surveillance is that the samples are tremendously, overwhelmingly bacterial. So how do you separate out the eukaryotic nucleic acid? And just in the last year or so, we've seen some very promising technologies that are able to extract eukaryotic nucleic acid out of the bacterial pool. And that would lead us, because in, for instance, malaria, even in fairly mild malaria cases, we've been able to detect nucleic acid from falciparum, from the parasite, in fecal matter. So this would enable us to actually understand what the distribution of parasites are and what potentially some of the resistance genes. You may know that drug resistance to the malaria parasite is another problem that has been well known in Southeast Asia. We are particularly concerned about the rising problem in South America, which may be related to the tragic situation in Venezuela. So with environmental sampling, do we have the liquid biopsy transplanted from that idea in cancer diagnosis into the public health space? I could hardly not mention the extensive work that we've been doing with Caliber and Scripps in the drug space. And I had many topics that I could have touched on over here, whether it be in TB and even in terms of other factors, such as this very troubling inflammation that kids have in many developing countries that really leads to the problem, we believe, of stunting. And even though you may refeed these kids, you are not able to relieve the stunting because of this intractable inflammation that they have. So gut barrier function is another thing we've been looking at with Caliber. But this relates to malaria. There's a compound called KAF156, which is a compound that Pete knows well, and the group over here at Caliber worked on it in a slightly different incarnation. Both Kelly and Arnab were part of the team that invented this KAF. Is the most promising molecule in the clinical portfolio for malaria at this point in time. But this is a different idea. So people found that a compound known as ivermectin, which has been used to treat parasites. You may treat your pets with it. It can be applied topically, for instance. It's what they call an endectocide. So it treats these ectoparasites that animals have and humans can have in the terms of things like head lice or scabies. Well, it's been found that actually ivermectin itself is toxic to mosquitoes who feed on blood of people who've had ivermectin. And ivermectin's got a relatively long half-life, so this effect of its insecticidal effect. So the idea being that the mosquito that might have malaria comes along, takes a blood meal from an individual who's had ivermectin, and that has an insecticidal effect. Lasts about a week. Well, the lifespan of a mosquito is 21 to 29 days. So a week is not good enough. We don't want a week, we want months. And with the collaboration over here, we've been looking at a class of drugs, fluralana, ifloxalana. These are drugs that have been used in the animal health area, do seem to have this profile that they may sustain insecticidal levels for up to 90 days, which is perfect in the setting of seasonal malaria in those countries that just have a fairly short malaria season, which could be just a few months, 3 to 4 months, and where this might be exactly what you need to block transmission. But you can imagine, now we need a compound that is incredibly safe and retains this tremendously long half-life. So it's a huge challenge to produce a compound that you would go into a population and be happy to have that be given without any kind of pre-diagnostic. I've given you some of the examples that I wanted to put forward in terms of this innovation for us to make a difference in the global and public health spaces. But there's another set of factors, which is how do we put together the ideas? How do we put together the groups with the various competencies? How do we bring together all these fantastic technologies? We read about them every day, but they are in silos. They may be funded by funders like NIH, by funders like us. How do we get that mass effort in a more effective, synergistic format? And I think 1,000 ideas are necessary over here. I don't think that we at Gates have got any of the final solutions, but we are trying a lot of different ideas. And many of them have been generated by folks within your communities who've come to us and said, let's try this way of working. I just want to touch on a couple of the things that we've been working on in terms of bringing together these various options, these new technologies, in a more effective way. So on the discovery side, we've a number of institutions caliber that we fund. Institute for Protein Design, David Baker's Institute, Stanford, Mark Davis in human immune profiling. He does a lot of really cutting edge immune profiling for us. Bruce Walker's Raygon Institute at MIT, which is working largely in the HIV and TB space. And there are many others. There's the Global Health Drug Discovery Institute in Beijing as well. So there are these entities which are funded and are doing fantastic work within their own areas. How do we bring them together in a network where there is an effective exchange of information and the technologies that one of those groups may develop, whether it's a David Baker nanoparticle, can be utilized by Caliber? How do we get those groups to work in an effective way? And we need a new model of funding. It can't be the 3-year cycle of competitive publication type of funding. Even the venture funding, which has got so many strings attached and got so many constraints, doesn't seem to be the only answer. So we need a panoply of solutions over here because we very much want to bring in the private sector, whether it be big pharma, whether it be biotech, and we want to be able to use the platforms that they've developed as well for this kind of work. And they've been open to that, but they need the format. So we are working on this hub of discovery institutions over here and would love your input into how we move forward with that enterprise. Translational science, in terms of that critical juncture from idea and research into the clinic. And we've been talking how important it is. And we've seen progress. I think there's been progress in the oncology area in particular. But I would say that overall, I'm somewhat disappointed that we haven't been able to put the resources together in the right way, that we have really effective translational science. And so we have to find different formats. We established about a year ago Bill and Melinda Gates Medical Research Institute in Boston. And they've been up there for about a year now. We have about 60 people. They are tasked now with building up a really world-class set of capabilities that go across the disciplines that you need in order to bring these ideas through into the clinic and work in very difficult regions. We located this in Boston because we hope to borrow tremendously from all of the innovation that is going on in the oncology area. So we think in much the way that oncology had its origins in some of its methods in the infectious disease space, we would not like to see the reverse transfer. We will gain back from oncology all the advances that made, whether it be liquid biopsies or other technologies that we can transfer back into the infectious disease space. Of course, there's regulation, and we've invested a lot of funding in this area, working both in terms of the global regulatory system, which is the pre-qualification system at the World Health Organization, which we have funded over the last many years significantly. And I think that there's been a tremendous improvement in their ability to reduce the lag. Previously, the problem was that a new innovation would be approved in the US or in Europe, and then it would be 5-10 years before that would be available in the global markets. So we've managed to get that down, I think, to a much smaller number in the space of vaccines, diagnostics, and drugs. We still face challenges in the area of vector control products, which are important for us, but that's the next frontier. But it's more than WHO. It's the countries that we are working on in sub-Saharan Africa. For instance, there have been investments in aggregation of capabilities in countries that have got very weak regulatory systems. How do they pool those? The East African Harmonized Regulatory Region now is 5 countries in East Africa that are pooling, each country having limited regulatory resources, but all told actually reasonable. SAPRA, which is the new regulatory system in South Africa, which is important for the whole swath of Southern Africa, is in terms of its ambition, is a huge, huge change from the previous system which was had over there. But there needs to be an exchange, and we've been involved in trying to transfer capabilities and intelligence from agencies like the FDA and the EMA, and have transfers across both into Africa as well as into other places like China, for instance, our work with the regulatory system over there, because for a different purpose. In China, we want to be able to produce things of high quality that we can actually deploy. What I'm most excited about on the regulatory front, though, is this issue of data science and the Office of Innovation that Scott Gottlieb announced recently. So we intend to do I think a lot of work, I hope, with that group in terms of how do you bring these new and fascinating and important data science tools to the regulatory system. So the idea is that we'd always dreamed about is a sponsor would drop their raw data at the door of the regulator, who would have great data science to examine it and decide whether it was worthy of approval in terms of efficacy and safety, and they would get the thumbs up or not in about 2 weeks. So is that an impossible dream? Well, you need to have a tremendous investment in a new capacity, which I think existing regulators, the FDA being at the top in terms of capabilities, do not have, even the FDA doesn't have. And we would love to see the emergence of that kind of capacity in the regulatory systems, which will infuse everything with a sense of urgency if we can get that right. So I hope that I've given you just a brief overview in terms of some of the work we're doing. And if any of you are thinking about where are the challenges that I would like to address for the next period in my professional career, I would suggest that if you want tremendously high challenges, the toughest challenges in the biomedical sphere, then you should apply your capabilities in the global health space. And I do want to really appreciate the invitation from the Scripps folks for inviting me down over here. And as I've mentioned, this is not just because they're an excellent group of scientists and doing excellent work with us, but because I think their hearts are in the right place. Thank you.